Welcome to Brain Ponderings. I'm the host, Mark Matson. Today it's really a true pleasure for me to talk with a longtime friend, research colleague, a chemist, biochemist who's an expert on the chemistry of oxygen-free radicals, where they come from, what they do, good things, bad things. And he has a lot of interest in brain aging and Alzheimer's disease and has made major discoveries concerning what happens in membranes in cells in the brain in aging and Alzheimer's disease and related to free radicals attacking the membranes and why that is occurring, how it is occurring, and what's happening, I guess, both upstream and downstream in the progression of the disease. So, welcome, Alan. It's really good to see you. Last time I saw you was, well, you invited me to give a talk at the University of Kentucky
Right.
in Lexington, where you were a professor for
50 years.
50 years and chair of the Department of Chemistry for some time period, I think. And
I was in the administration of the chemistry department,
ah,
but not chair. But I have been associate vice president for research
ah,
quite a long time, maybe 15 years.
yeah, and
But
you
retired January 1st this year. So
And you were where I met you, I got recruited to the Sanders Brown Center on Aging there, one of the first or the first maybe NIH funded Alzheimer's disease research center, one of the first.
yep,
It was headed by the late Bill Marksberry, who was just a neuropathologist, world renowned, one of the
absolutely.
best people I've ever known, the best pathologist. And he Yeah, it was amazing. He just let me do what we wanted, gave me encouragement, gave me a few ideas, including, well, and you gave me some ideas, and I guess in this conversation we'll talk a little bit about our collaborations because it's kind of an
Absolutely.
important role. Okay, so you're up in Maine now. You said that you recently retired after 50 years, and you were born and raised in Maine, right?
True. Yeah, up and further north than where I am, another 100 plus miles north, and even then, that's only the center of Maine. So there's still equal distance further till you get to Canada if you go north. So it's a very big state.
Yeah,
When
there's this kind of extending to the northeast, this big section that, and
right,
most people who go to Maine, they come from whatever, Boston, or they
yeah,
come up
absolutely,
there and
yeah,
then they hit Portland
yeah,
and
Old
kind of
Orchard Beach, that sort of thing.
Bar
Just
Harbor and stay
yeah,
around
Bar
there.
Harbor.
Yeah.
Yeah, exactly.
Yeah.
Yeah, that's Maine is that's probably its largest industry is tourism.
Yeah. We've been up there. It's one of my daughter's favorite places to go. It's about, well, what, I guess 10-hour drive. It's a 10-hour drive. We've rented a house, stayed for a week up there. One year we went up in the summer, and here in Maryland, me and my son have like foraged for mushrooms, right?
Right.
And there are some mushrooms, but we went up there and we went on this trail, and within an hour we found probably 20 different kinds of mushrooms.
Oh my gosh,
Yeah,
that's wonderful.
yeah. Only one we were confident was edible.
Yeah, well, yeah, you have to be careful.
Yeah.
My good friend Barry Hallowell has made a career of ergothioneine, and which is deeply found in mushrooms, and he has showed a slowing of MCI into AD by uh, getting purified. He has a company that made purified ergothioneine, and in his
He
clinical
did a
trial,
clinical trial?
he did, yeah, that's a small one,
Yeah, okay.
30 people, but he showed less rapid movement towards dementia if they just load up on ergothioneine.
Are
And
they going to get funding for a big study?
They tried. So I've been on the review committee for the Ministry of Health of the government of Singapore for
Ah,
about 10 years. And they see grants the minimum $10 million and maximum $25 million. And he put in for a $25 million grant for um, a clinical trial, but unfortunately it wasn't funded. So,
yeah.
but it not without, I mean, not because of anything wrong. They just thought he needed to have a lot more people. And when you start doing, as you may know, doing clinical trials is very expensive and
Yeah, they're usually supported by,
by industry,
by huge
yeah,
pharmaceutical companies.
which are all present in Singapore. So, but ergothioneine is still he's still having to educate pharmacological companies about um, why this is important, but
Yeah.
it sure is. So um,
Okay.
anyway, he's doing that. Anyway, yeah, I'm I am retired after 50 years. Uh, Part of that time, you and I worked really closely together, and you're the person who helped me first when I was walking down Rose Street back to chemistry from Sanders Brown, and I almost got run over by a car because I was thinking so deeply about why in the Alzheimer brain are there so many proteins and lipids that are shown to be different? Why isn't there just one, like in sickle cell or something? And
Hmm.
and I said, oh. What if there's like a free radical that whatever it hits, it's going to change the structure and would do that? So I turned right around, went back to your lab, and I said, hey, Mark, do you have any of that peptide that accumulates in the Alzheimer brain? And you gave me some. And I took it back to chemistry, put it into a solution that had a spin trap in it, put it into the EPR machine, and up came a signal. And I said, holy.
Can you explain what okay, go a little over the basics of free radicals and then explain what this method EPR is that you can.
Oh, yes. So everybody knows the word NMR and MRI and all that sort of things, but EPR measures the magnetic properties of electrons. And in free radicals, a free radical is defined by an entity that has an unpaired, at least one unpaired electron. The molecule oxygen has two unpaired electrons. And so it can double react once and then a second time. That's in fact important in lipid peroxidation. And so now how do you detect free radicals? You detect free radicals by putting them inside a gigantic magnet. I mean, huge amounts of magnetic, much more than NMR. And then under microwave frequency, so very short wavelength frequency, and relative to NMR. NMR is not so different than radio waves in some sense. So it's a long wavelength, but for EPR, it's very short to do this. Anyway, this machine will detect free radicals and it will make a signal come up. And that signal can be split. And in my PhD at Duke University in chemistry, I worked with the person who had developed the theory behind splitting of the Zeeman line, that every electron can make one line. But if it now interacts with other nuclei, like nitrogen, for example, not nitrogen 14, but nitrogen 15, and then it can work on it. Anyway, it can work with all kinds of nuclei and be split by their magnetic moments. And so he developed a theory of that. And I thought, well, this would be good to go study quantum mechanics with him. And he said, well, yeah, you could do quantum mechanics, but they don't have good chance of getting a job when you're done. He said, but I worked with Hardin McConnell and as a theorist, He was a theorist, Hardin McConnell, at Caltech for my PhD, and he's now developed this method called spin labeling, in which and he said it's being applied to something called a cell membrane. And he said, I don't know what a cell membrane is. And so he said, but look, we're here at Duke. You ought to be able to figure out from talking to people, and the biochemistry department was made up pretty much of everybody in the National Academy of Sciences. So every day I could go to the biochemistry department and be made a pain in the ass of myself because I just tried to learn all this. And it was just fascinating, stupid fascinating. So anyway, my PhD advisor says, "You teach me cell membranes, and I'll teach you magnetic resonance." And it was so intellectually stimulating to
Yeah.
do, and everything I touched worked. And so I earned my PhD in 2.5 years, and it was really
Well, and then you used your expertise in chemistry, which I'm weak in some ways. But my background is in like cell biology, how do cells
sure,
work?
yeah.
And
And I didn't know jack about that when I was a graduate student.
yeah,
And until then I said, I've got to learn this. So I went to the same class that the medical students take for biochemistry. And I sat in on that class and as a audit sort of thing. And we covered a famous biochemistry book in one semester, the whole.
Leninger, was it
It was
Leninger?
Leninger,
Yeah, yeah,
absolutely. And it was very much oriented towards proteins and enzymatic sorts
yeah.
of things. That, and I said, where has this been that I didn't know anything about this? So it just opened up my mind to all sorts of things. And I said, oh gosh, I bet that this can be applied to diseases like neurological diseases. There's all kinds of things going on in the brain. And I bet we can figure this out by using chemical principles like this. And really, that was the genesis of my getting into research in neurological
Right.
disorders.
So you came over to my lab and I gave you some of the amyloid beta
Yeah,
peptide and you put it in the EPR. What did you find?
we found a three-line signal showing up. And I'm saying, holy smokes, that's being split by the nitrogen that's in the spin trap. So you get three lines, and a spin trap is not a free radical, but it traps a free radical, and then becomes
Changes.
a paramagnetic molecule that you can see the signals for, and it makes a lone pair of electron. It makes a lone electron on the nitrogen because oxygen, it's bound to oxygen, and O bond. O is more electronegative than N. So it draws the electron density towards itself, leaving an unpaired electron on nitrogen, which then splits this signal into three lines. And yeah, so that was the beginning. And you might remember my graduate student, Kenny Hensley.
Yeah, yeah.
And Kenny, kid who grew up in rural Appalachia, poor as a church mouse, and but smart as a whip. And when that signal appeared, I said, Kenny, there's your dissertation right here.
Yeah.
So here's what you're going to do. Go for X, Y, and Z and just don't stop. And he graduated in 3 years and had 26 papers published. Can you believe that?
And
That
the
that
first
guy,
one though, so you saw this signal
yeah.
indicating that the peptide, when it's just in a physiological solution, it generates some free radical.
Somehow, yeah. Now there could have been some metal contaminant in the solution. I tried to remove all of that using deferoxamine and other things, and I purified the trap 3 times. You precipitate the trap out, and then you redissolve it, and then you
Huh.
precipitate it out, and the trap showed no signal. If you just do the trap by itself, there's
But
nothing.
then you also didn't you remove oxygen and then you didn't get it?
Yes, if you remove oxygen, you get nothing.
Yeah.
Absolutely right. So I knew oxygen had to be part of it. And I'm helping. Chris Norris at UK now. He wanted to know of a key interest in an experiment. How could he prove this or that? And I said, just put nitrogen through your buffers for a half hour. Just blow it through it. It will drive out the oxygen. And now, if it's a free radical reaction that you think is happening, you should be able to prevent it. And so he's going to try that. And his first experiments suggest it's right, but he's still got to
And then
do it.
so the first paper was a PNAS paper, or the big paper, and it raised a lot of interest, controversy.
Oh my gosh. I presented the work at the Society for Neuroscience meeting, as you know, a huge meeting. All of the big names in Alzheimer came to hear this talk, and mine was the only paper at the time, the only abstract that mentioned free radicals and Alzheimer and all that, and A beta. So I presented this. Every one of them lined up and said, "That can't possibly be right," and it just can't be. And so, my neck was stuck out there pretty far. But I have always believed that if you're not willing to bend the paradigm a little bit, you're never going to learn anything new. And so I said, well, I've had three of my graduate students independently verify this. So I'm pretty sure it's real. So the next year, I come back to the neuroscience meeting and again present the work. And then these people say, well, we knew Butterfield was right all along, and we just we confirmed everything. And so, yeah, we knew that. So, uh, you know, it, for that 1 year, my neck was pretty far out there, but by the end of the year, everybody bought into it. So that was good.
So then in Alzheimer's disease, the amyloid's accumulating outside the neurons.
It
And
is
and
as a fibril.
yeah.
Yeah,
And
mine
we
is
we had as you know, at that time we were doing a lot of studies in cultured neurons,
correct.
both from embryonic rats and some human neurons. And we'd found that if we expose, if we expose neurons to very high levels of aggregating, high concentrations of aggregating amyloid beta peptide, that can kill them. But if we expose them to lower levels that kind of don't seem to have any obvious adverse effect, the neurons are still very vulnerable to being killed by excitotoxicity. So when they're being stimulated, okay, so then that's when we really got into the collaboration because,
Right,
and then we had all this evidence from my group that there's a disruption of calcium regulation, probably at the membrane.
right.
So then we had this collaboration that really took off. So can you talk about what we did there?
Yes, yes. We did lots of studies, not only in cell culture, but in various mouse models of Alzheimer's disease. That I happened to be at a neuroscience meeting when uh, Dorothy Flood, is that her name? Dorothy somebody, I've forgotten her name.
Yeah,
She worked for a company and she had built the uh, knock-in mouse, not a transgenic, but a
yeah.
knock-in mouse having the transgenes or having the mutated genes for APP and tau, and built that mouse, and it was just amazing. Anyway, what we did with that is we examined A beta, that we worked with a company that so we worked with the A beta 40 and the A beta 42 molecule. And sure enough, we saw that there were cell death being produced, but also lots of free radicals that are being produced. And we could detect that by immunochemistry because the free radical signal of a protein are things like 3-nitrotyrosine is the mark of what happens when nitric oxide and superoxide react. They make a molecule called peroxynitrite.
So yeah, that's two free radicals interacting.
That's two free radicals. That's right. NO is, how do you know NO is a free radical? Count the valence electrons. Nitrogen has five, oxygen has six. It's an odd number. So there's got to be one left over because every bond takes two electrons. So NO is a free radical, and superoxide free radical is by definition a free radical. And a radical-radical recombination is the fastest reaction known, and or among them anyway. And so it makes this non-radical peroxynitrite, and there's through a series of chemical reactions that ends up uh, making a NO2 group on the three position of the tyrosine, uh, the phenol ring of tyrosine.
Mhm.
So it's right adjacent to the OH, and it sterically blocks the OH from getting a tyrosine kinase to be able to work on it.
Yeah.
And so all of the signaling pathways that involve tyrosine kinase,
Yeah.
uh, they're screwed up because of this, and that could certainly contribute to cell death. Um,
And some good examples of uh, uh, proteins that the phosphorylation on tyrosine affects their function is growth factor receptors, like brain-derived neurotrophic factor or nerve growth factor.
Good point.
Yeah.
And all that fits in, by the way, in some of your brilliant work on what happens on fasting, and also you and Carl Kottman had worked together on antioxidant diets and seeing what would happen there, and exercise. And he found that brain-derived nerve growth factor was a product of exercise and these free radical diets.
Yeah.
I mean, antioxidant diets that were fed well, these were beagle dogs at the
Yeah.
time, along with Liz Head working. She's the one who really did the work and developed the field. And um, anyway, it's just amazing. So the other marker for free radical damage to a protein is called protein carbonyls. So I wrote at your invitation, actually. You were editing a book on biology of aging, I think, and Earl Stadman and I wrote a chapter, and he was Mr. Protein Carbonyl.
Yeah.
He developed this as a marker of aging, and no matter what species he looked at in the and what other organ, like a liver versus a brain, whatever. Uh,
He was known at NIH as a great mentor. He mentored Nobel Prize winner.
three.
Yeah, three Nobel Prize winners.
Yeah, as a matter of fact. Yeah, he was also a mentor to me in many ways. He helped us get our first PNAS paper published because he was
Yeah.
at those days the people could send it in as long as they had good reviews from other NIH National Academy of Science people.
The kind of, I mean, your initial finding with the peptide generating free radicals is I kind of make an analogy somewhat with Stan Prusner and the prions, although I think his even met with more resistance than
Oh yes,
your,
yeah,
yeah.
yeah. Ours definitely, and also if you, I know we want to talk about lipid
Yeah,
peroxidation,
we do.
and I will, but let me just finish up maybe about this. If
Okay.
you look at A beta uh, amino acid sequence, there's only one amino acid in A beta that has redox chemistry in it, and that's methionine. And so I said, well, if the free radicals are happening here, methionine got to be important. And so we did all kinds of experiments with methionine and showed all kinds of ways in which this was important. But finally, with the help of Dale Bredesen, he built us a mouse in which it was a J20 mouse. It was Swedish and Indiana double mutant APP, and then added a third mutation of changing the methionine to leucine. that is in a which is in the A beta 42 sequence. And we grew up that mouse to a year, uh, looked at the brain, and there was no oxidative stress compared to the litter mates that had these that didn't have this mutation. Um, so I said, come on, there it is in vivo in a mammalian model. We had already previously done that in C. elegans, but you could criticize that maybe it's not mammalian.
Yeah.
So here in a mammalian model, there it's clear there's methionine is key to this whole process, and that if anybody wants to look up the mechanisms of all this in my paper in 2024 and physiological reviews, I really lay this out perfectly about how methionine plays a role in this. And
And
it involves,
I'll put a link to that paper in the description section.
oh, thank you.
Yeah.
And this all is related to lipid peroxidation. This is the point where we could talk about this. So I don't know how much chemistry I should say, but so let me try to be very not too technical, but just give the big picture here. In the case, so we have to do one thing else is that um, um, let's see. For some reason, I'm forgetting his
Well,
name.
I was maybe
I
start
know.
with the composition of the membrane and
Yeah, that's right.
particularly, neurons are enriched in certain
Yes,
fats.
polyunsaturated fatty acids,
Yeah.
right?
Yes.
Yeah, arachidonic acid especially, but docosahexaenoic acid as well. And but the arachidonic acid is particularly susceptible to oxidation and peroxidation. Peroxidation meaning more than one oxygen is in the product that happens. So it becomes a peroxide when you're done, and actually a hydroperoxide. So yes, if you have a free radical, and mind you, free radicals are usually so reactive, they cannot be formed outside the neuron
Right.
and affect something inside the bilayer. By far, they would have reacted well before they could ever
Except
be.
maybe for nitric oxide, because
Well, that's possible. Uh, it,
it's a gas actually.
yeah, it's a gas.
Yeah.
It might that I should say I should say that in the context of reactivity, nitric oxide is not very reactive except in another radical around it.
Ah, okay.
Okay,
Yeah.
so but if you had something like superoxide,
Right.
right? Um, or even worse, OH dot. OH dot is a very, very reactive free radical.
Hydroxyl.
Yeah, hydroxyl free radical formed in many ways by iron,
Yeah.
reactivity with hydrogen peroxide,
Or
and,
copper.
copper 1 or iron 2. It has
Yeah.
to be the reduced form of the
Yeah.
ion. And yes, it will make OH dot, and OH dot will react with anything almost. It is so powerfully reactive, and including the nature of the unsaturated fatty acids in lipids in the neuronal membrane. Now, let's think about that. Most people know the word unsaturation means two carbons are connected by a double bond,
Right.
right? And then something like arachidonic acid, you have four such double bonds, each separated by a carbon that's not connected to the double bonds. Oh, well, it is in one sense, but it's not,
Sure.
it's, but that carbon itself is not have a double bond on it. That's called an allylic carbon. And an allylic hydrogen is bonded to one allylic carbon. And now, organic chemistry, if people remember that when they took organic chemistry, allylic bonds, so a hydrogen bonded to an allylic carbon, are very labile. That bond is very weak. So along comes a radical. It's going to abstract an H and one of the electrons in the double bond of the bond between hydrogen and carbon. And that H is going to go back to wherever the radical was that came and got it.
Uh,
And and then you're left on carbon with a free radical because
so
it
you've got
has
a
an
chain reaction type thing.
exactly. And now, how does that happen? A chain reaction happens because oxygen, O2, is present within the bilayer. How do you know that? Because it's a homonuclear diatomic molecule and is therefore have zero dipole moment, meaning it is completely nonpolar. So it's soluble in the
Yeah.
greasy material of a lipid bilayer. And it has two unpaired electrons in it. So all of a sudden you see a carbon with a radical on it, a free electron, and oxygen and some people argue at level of millimolar. I mean, very, very high levels of oxygen
Huh.
and certainly micromolar. So very, very high amounts of oxygen. And it can then do radical recombination to make a O, so a a the radical binds to this carbon and makes a CO bond, as it were, and um, and uh, then then we have this oxygen can also bind to a different uh, allylic hydrogen and then produce another reaction. The idea is there'll be another oxygen that will go to this piece. Okay, so the first bond is carbon, oxygen, oxygen, dot. And now that dot on the O of the second O of COO dot, that swings around and grabs another allylic hydrogen and becomes COOH. But on the carbon it just took the electron of the hydrogen from,
Yeah.
there's now a free electron. That's what you mean by a chain reaction, because that will continue to happen as long as there are allylic hydrogens and oxygen present. And that's why when you devoid the system, you make the system devoid of oxygen, you can't make lipid peroxidation work. And
So you can have essentially one molecule, one hydroxyl radical molecule or one A beta molecule can wreak a lot of havoc. And
tremendous. And where the radical comes on A beta is a S plus dot radical of the methionine. That
that's
we've
interesting.
proven that. And so now that's right in the middle of the bilayer, shouldn't be a plus charge in the middle of the bilayer. So it immediately reacts, that electron, the S dot, can start this reaction onto the allylic hydrogen. Now, how do we know there's an S plus dot radical on methionine? Uh, it, because A beta is in the bilayer as a small oligomer, dimer, tetramer, trimer maybe, and uh, a giant fibril, there's no chance in the world it can be in the bilayer.
Right.
So it has to be small. And uh, when it's when A beta is solubilized in the bilayer, it's like a protein, and almost every protein solubilized in the bilayer adopts the secondary structure of an alpha helix,
Yeah.
including A beta. Then alpha helix, every fourth amino acid interacts with each other. So on so methionine is residue 35. So we reasoned if we put leucine is a leucine, not leucine, it's isoleucine at 31. And so we substituted we made the peptide with proline in the 31 position.
Okay.
Proline is a helical breaking
Hmm.
amino acid.
Hmm.
So it won't form an alpha helix. When you do that, And then you add this to the neurons in culture, zero cell death, zero free radical. Isn't that incredible?
Yeah.
So that tells you that this has to be the reaction. And the reason that happens is the carbonyl, so the amide bond in a peptide bond has a CO or yeah, CO bond as part of it. Well, that O is right within 4 angstroms of the sulfur, which has it's in the same group as oxygen, right? So it has two pairs of two pairs of electrons, like oxygen does. Also, oxygen has two unpaired electrons. But anyway, sulfur here has these two electrons, and oxygen, being far more electronegative than sulfur, is drawing the electron density away from the sulfur, meaning a free radical of some sort in the bilayer that can either be oxygen or I think it could be copper.
Hmm.
But copper 1,
Yeah.
actually copper 2, it becomes copper 1. And anyway, we had the idea is that this can interact. Uh, so the electron density is taken stretched away, meaning that these two electrons of the lone pair are less tightly held to the sulfur nucleus, which has all these protons in it. So a radical can come and grab one of those, and that's what makes the uh, S dot plus radical. And that S dot radical can immediately pull an allylic hydrogen off the carbon.
Okay.
And when that happens, you can come to SH plus and a free radical on carbon. And then the whole thing with oxygen takes place. And it just goes and goes and goes. And it just constantly does that. Now, what happens to the SH plus on the methionine is that it has a pKa, in other words, a tendency to lose the proton that's bound to it, of -5. pKa is -5. That means it's very labile. It'll fall right off. So any base could be anything, even water, can take that H plus off. And what do you have left? You have methionine. So you start with methionine, it goes through a reaction, it comes back to methionine. That's what you call a catalyst. So it's both a catalytic reaction and a chain reaction that is leading to this production of lipid hydroperoxide, COOH, is the modification of that original lipid, carbon. And that molecule, the COOH on the acyl chain of like arachidonic acid, that will immediately break down into HNE. Now, it takes about 10 steps, but it makes what is called 4-hydroxynonenal.
Yeah.
So nonenal means 9
Yeah.
carbons, and ene means there's a double bond in it, and al means it's an aldehyde. So it's aldehyde on carbon 1. So that means a double bond O and an H. And then you have carbon 2 and carbon 3 and carbon 4. And the between and then all the way to 9 carbons. So 1, 2, 3, 4, I've just drawn myself to see. And then you have it's an ene, and an ene, the double bond is between carbon 2 and 3. So you have, let's just see in your mind, carbon 3 double bonded to carbon 2, single bonded to carbon with a double bond O and an H on it. This
So
is
this amyloid beta peptide, it's kind of intercalating somewhat into the membrane,
it has, yeah, absolutely has to be there in order to
it's
have this reaction occur.
through this free radical mediated mechanism. It's causing essentially cleavage of these lipids, this arachidonic acid, for example, and then this 9 carbon piece is
Is
released.
Yes, it's nine carbon. It's right. It breaks down the oxidized lipid, the peroxidized lipid, and results in a nine carbon aldehyde that has a double bond between carbons two and three. Now,
which I know well, and you know well, and Bill
yeah,
very knew very well.
but on carbon four, there's an OH. Okay,
Yeah, yeah.
and so it's 4 hydroxy. So 4 hydroxy, two nonenal. So the double bond
And
is
then with that chemistry, this 4-hydroxynonenal can move easily. It can move in the membrane, it can leave the membrane, it can move within different cell compartments.
You are absolutely right. Earl Stadman was the first person to show that.
Yeah,
It can go from the bilayer even to mitochondria
yeah.
and damage some of the lipids there.
All right,
And
I even saw. Talk
so
about what happens when this 4-hydroxynonenal
okay, so
with
this is
protein.
this is really important
Yeah,
because in carbon number three, so remember carbon number one, double bond OH, that's the aldehyde part. Then the single bond to carbon two, but a double bond to carbon three. And a single bond to carbon four, but carbon four also has an OH group on it. Now, OH, remember O, more electronegative than carbon, it's going to draw electron density away from uh, the uh, electrons on carbon three. It's going to be drawn towards carbon four. But in addition, you might remember the technique that's called the inductive effect in organic chemistry. And the C double bond C in carbons three to two, that double bond can undergo resonance. That double bond can switch between where it is to carbon one, carbon two to one. And that makes the double bond O become a single bond with a negative charge on it. And then when so that makes carbon number three, that's the point. Carbon number three is electron deficient. Some of it has been drawn, the electron density by the OH, and the other by resonance. Carbon three is deficient, so now that means it can be attacked by any molecule that has electron density on it. For example, the S double bond of methionine or any other protein. For example, actually be easier, not methionine so much, but cysteine.
yes, this
Cysteine has the SH group, and histidine has a lone pair on the nitrogen, um,
and lysine.
and lysine. Right. Lysine is less reactive because it's got a positive charge and has to get organized there. But for sure, cysteine is probably the most reactive, and histidine also pretty reactive. And so those sulfur with a lone pair of electrons on it on cysteine just has a side group of CH2SH. Oh my goodness, here's this carbon with no electrons on it, and here's my electrons. I'm going to attack this thing, and it makes a covalent bond.
Yeah,
Now, when it makes a covalent bond, it immediately changes the structure of the protein.
yeah.
And every chemist learns function follows structure. Change the structure, you change the function. And when we invented a method called redox proteomics, we were able to identify which proteins in the Alzheimer brain or NCI brain or control brains were oxidized by HNE binding like this. And we measured the activity of those proteins if they're enzymes, and to all but one, which was amazing. Anyway, these all the proteins we looked at, except one, lost activity when HNE bound to it. And
And that followed. So you did most of that after I left Kentucky in
yeah, I did. Yeah,
2000. But
yeah,
before that, so I had a graduate student, Bob Mark, and then Jeff Keller, who I kind of co-mentored,
yeah.
PhD. And so, we could purchase 4-hydroxynonenal
Yes,
from company.
right.
And So we just added 4-hydroxynonenal to the cultured neurons, and then we found it made them very vulnerable to excitotoxicity. And we found that Bob found that it impaired the function of the ion pumps that pump sodium and calcium back out after neuron
Absolutely.
fires.
Right.
And then I think both you and my lab found that it impairs glutamate transport. So you
Yeah.
get more glutamate accumulating outside the neuron and
Right.
exciting.
In the synaptic space.
Yeah.
Right. And absolutely right.
And also glucose transport. So,
Absolutely.
we had
Glucose.
all this evidence from cell biology that this
This
aldehyde that's you found that's generated in response
absolutely
to A-beta.
exactly right. And this all ties very nicely into your work and
Yeah.
my work in our lab. Uh,
And the kind of the a global view of what's happening in the
absolutely.
brain. A, a scenario that involves amyloid beta peptide, but doesn't necessarily have to involve it and can occur in other situations where there's no amyloid around because there's many ways to cause lipid peroxidation.
Correct.
Yeah.
Yeah, that's right. And yeah, including, by the way, iron related
Yes.
lipid oxidation in a process called ferroptosis. It
Yeah.
can certainly, in fact, lipid peroxidation is the major driving force of the whole process called ferroptosis.
Yeah.
And without lipid peroxidation, it would be hard to imagine ferroptosis working. There are different ways to initiate various processes, but ultimately, lipid peroxidation has got to be a player. Otherwise, it's not going to work.
I often, with my background, it's kind of broad, and my bachelor's degree was in zoology, so I've had a strong interest in evolution.
Sure.
And then, I had neuroendocrinology and developmental neurobiology. And so it's thought that the earliest life on Earth arose, the first cells, probably bacteria-like cells, in essential, essentially underwater volcanic vents in the ocean,
Right.
in the deep
Right.
ocean. And that environment is very toxic. There's a lot of iron, hydrogen sulfide,
Right.
and then so all life evolved. Iron's very toxic because it's a very potent inducer of lipid peroxidation.
Correct.
And so organisms, cells, and then organisms evolved multiple ways to to prevent even accumulation of very low levels of this free Fe2, the ferrous iron.
Right.
And so there's these a lot of iron binding proteins. Hemoglobin's one, but uh,
Yeah.
ferritin,
Everything. Yeah.
transferrin,
Right.
etc.
Right.
And however, there's quite a bit of evidence that maybe with aging and more so in Alzheimer's disease, there's somehow more free iron and somehow, leading to the kinds of lipid peroxidation mediated
Right.
pathway.
And that's absolutely so. So we have identified proteins that are oxidized through the metabolic processing of glucose, and
Ah,
including uh, ones that are involved in the mitochondria. And so, if
yeah.
that's that case, then you can imagine the heme proteins being damaged in that way to release iron. But also, in other people have shown that when ferritin or transferrin or some other protein that has iron is degraded by by the lysosome. So it's taken up
Oh,
by some reason, part of a cell taken up and degraded, then the lysosome breaks these components down into the smaller units, including iron, and it's going to spit it out, as you've seen in nice pictures in biochemistry books. And it's going to recycle the iron, but as free iron too. and there all of a sudden is unbound iron. The other thing of
So this make there. I don't know. No, this is very interesting and important. So as you know, yeah, Randy Nixon did
yeah,
a lot of
yeah,
interesting work on
right.
lysosome dysfunction, and we'd shown that there's calcium dysregulation at the plasma membrane. He has evidence there's calcium dysregulation at the lysosomal membrane, but maybe in both cases, well, we know for sure that there could be, radical mediated processes upstream of those problems in the lysosome as well as at the plasma membrane.
Well, and indeed, I gave a seminar over at his place in New York,
Yeah.
and um, I had already identified by proteomics the V0 ATPase that pumps protons into the
Ah,
lysosome to keep the pH low to allow those degradative enzymes to work. And because it was damaged by HNE and carbonyls as well, as if I recall right, then because of that, it doesn't function. So all of a sudden, you can't make the lysosome
yeah.
produce as much lower the pH to a significant amount to activate these enzymes. And now you have all kinds of problems associated with the lysosome.
Yeah.
And
Toxic garbage accumulating in this.
accumulates, absolutely right. And well, I use the word detritus.
That's a good word.
That's
Or schmutz. Schmutz is another one.
oh gosh, I'm trying to remember if there's another word in Yiddish, but I have a wonderful friend on our camp road in Maine who's Jewish, and he loves to talk to us in Yiddish sometimes, just so to teach us words that we never heard of before. It's pretty cool. Anyway, we have yeah, we found also related to all this is we identified by probing every step of the activation of mTOR C1 that when mTOR C1 gets activated, then um, you have two things happen. One is you can block autophagy.
Right.
That will stop. Now, again, then detritus builds up. Um, and you when mTOR C1 is activated, uh, by radical A beta can lead to that, by the way, directly. Then what you can see is the phosphor because yeah, that's what I want to say. Activated mTOR C1 is a kinase, and one of its substrates is P7OS6K.
Oh, yeah.
And and that molecule becomes phosphorylated by the mTOR C1 kinase. And when it is activated, when it's phosphorylated, it is a kinase. It becomes a kinase. And one of its substrates is the insulin receptor substrate 1, the very protein that comes off the insulin receptor once it's bound by insulin to then direct how is it going to go to either the MAP kinase kind of pathway or a different pathway. And but it's it when P70 the activated P7OS6K reacts with IRS1, it binds to an inhibitory site of IRS1,
Yeah.
meaning you can add all the insulin you want to the receptor, but that signal is not going to be transduced. And that is a biochemical signature of insulin resistance.
Yeah.
And which we know is a major risk factor for Alzheimer's disease.
Yeah. Oh, yeah. Absolutely. And this, so you mentioned that, well, obviously diabetes is defined by elevated glucose levels and impaired ability of insulin to stimulate removal of
Correct.
glucose from the blood. And then you commented on glucose itself as a can be a source of free radicals. Well, I guess it is. It's gonna. There's multiple ways that can happen. One is just kind of the healthy way, I guess, is that it goes into the mitochondria and
Right.
is used to produce ATP, and you get superoxide being produced. But
Yeah, the
then,
whole Q cycle is basically two half reactions of involving free radicals.
yeah,
And that's the lipid electron carrier, right?
yeah,
So as opposed to cytochrome C is a water soluble electron carrier. Yeah, absolutely. In a good way, those are good ways free radicals react to with different substances. So that's right. But when you have proteins that are screwed up in mitochondria because of other free radical damage to them, and the biochemical modification of these proteins because of either HNE or carbonyls or nitrotyrosine, then you get in real trouble. One of the good examples of this, by the way, is in the pathway involving heme oxygenase to try to get rid of hemin and
yeah,
all the heme stuff that is broken down in mitochondria uh, occasionally. Well, when mitochondria are disintegrated, right? You got to get rid of that heme. And so heme oxygenase produces biliverdin,
yeah.
and biliverdin has an enzyme called biliverdin reductase that makes it into bilirubin. But it turns out biliverdin reductase, we were the first to show it is highly nitrated on tyrosine and doesn't work.
Yeah,
So
Saul Snyder did a lot of work on that.
yeah, I'm sure he did. Also,
Yeah.
there's a lady, I can't quite remember her name, at Rochester, who did a lot on biliverdin reductase.
Okay.
And anyway, this it turns out biliverdin reductase also influences IRS1, and insulin signaling is really in some ways controlled. We've published quite a few papers on that
Yeah,
recently.
early on, the people were skeptical about whether whether neurons responded to insulin. And they do, and there is insulin
Sure.
resistance in neurons in Alzheimer's. And I think, and a number of other people think who've been working on this that, okay, there's a lot of evidence that glucose transport in neurons is impaired in
Right,
Alzheimer's disease. Even you go uh, radio labeled glucose PET imaging, and cells aren't taking up
right.
glucose. And then, but we think that there's still, at least early in the disease process, neurons that can still be alive and can use ketones
Yes,
because that pathway seems not to be, it doesn't seem to be a problem in the neurons taking up the ketones.
right. Yeah,
Uh,
this is exactly. That's a very good point. And this is what a baby does when they're born.
that's true.
They live in on ketones, so from the mother's milk,
Yeah.
right?
Yeah.
They don't really engage much in glycolysis or any of that. So, but at some point, that switch takes place and goes from there. Yeah, pretty interesting. Absolutely. Um,
I wanted to mention one other thing while it's on my mind. Okay, so this 4-hydroxy non-enal can covalently bind to cysteine, lysine, histidine residues.
right, and change the conformation of the
Lysine is
protein.
very interesting from a standpoint of enzyme function and signaling. We worked on sirtuin 3, which is a protein deacetylase in the mitochondria. That means it removes an acetyl group
Right.
from lysine residues. And we and others have shown this mitochondrial enzyme. It's very important for normal mitochondrial function and for protecting cells against a variety of stressors. There's other SIRT2 and SIRT1, which is seems to act mainly in the nucleus. Okay, so one thing I've always wondered, and someone it hasn't been looked at a lot is so presumably if a say a protein substrate of sirtuin 3 is modified on the critical lysine residue by H&E, one I would think intuitively then if it's a well, I guess there's two questions. One, if it's acetylate, does that affect covalent modification? And two, if it doesn't, then if it's modified by H&E, can it then not be acted upon by the deacetylates?
Correct. Yeah, the that last question is pretty straightforward, right? Because
Okay.
you have a carbon sulfur bond formed
Yeah.
in when H&E binds to to a molecule. The carbon from the H&E and the sulfur from cysteine or something. Now, in the case of lysine, same idea. The carbon nitrogen bond is a very strong bond. It's very unlikely, unless something drastic happens, that the deacetylase is going to come and remove that bond and be able to put
Yeah.
acetic acid, or I guess acetate, onto that. And so no, I don't. I think once H&E is there, this bond strength is so strong that
Yeah.
of a carbon nitrogen or carbon sulfur, and in the case, yeah, so in lysine it would be the nitrogen, and that's that's pretty tough to break.
Yeah.
And
Yeah,
now
but I guess,
the first
so you
one,
could imagine, have mucking. So one of the most robustly affected enzymes in the mitochondria by deacetylation is SOD2, manganese superoxide dismutase, which I think is probably the most important antioxidant enzyme in the cell.
yeah,
And and so when it's deacetylated by SIRT3, it increases the activity of the SOD2. And so if
oh,
that's impaired by H&E, then that's really going to exacerbate the
yeah, actually, the biggest change in SOD2 is nitration.
ah
So it absolutely does that, both in AD models, but also we studied a process that patients call chemobrain, but basically it's chemotherapy-induced cognitive impairment, and SOD is definitely affected there. And that's pretty important. That's also a free radical process. But by agents, by the way, just as an aside, the chemotherapy agents can't cross the blood-brain barrier, and yet they cause changes in the brain. How does that work? And the answer is it upregulates TNF alpha in the periphery,
mm.
and the TNF goes in by
So
uh,
inflammation.
receptor mediated endocytosis, and then it activates inflammation, yes, in the neuron, and ultimately kills it. And
So
that
let's kind of step back and summarize what we've talked about in terms of kind of a sequence of events that you think are occurring in during the development of the pathogenesis of Alzheimer's disease.
Right.
And then after we kind of summarize that, you can talk about potential interventions to either well, from a preventative approach, risk reduction, or a therapeutic approach.
Okay. So starting at the beginning, and remembering that similar proteins, almost not every time, but a lot of similar proteins are oxidized in the MCI brain. There's no dementia in MCI brain, but these same proteins are oxidized. And
Well, you could say there's kind of early dementia, maybe.
you can say there's memory issues, there
Yeah.
are some cognitive changes, sure. but it's
But
not
that's comparing someone with mild cognitive impairment to someone of the same age who does not have mild cognitive impairment.
correct. Correct.
Yeah, yeah.
And anyway, yes, so these proteins are identified by us in using redox proteomics as having HNE or nitrotyrosine or carbonyls bound to them. And so this is so in other words, oxidative damage is an early event,
Yeah.
absolutely early event. There is, we couldn't demonstrate it ourselves, but Mark Lovell has done some work. We couldn't quite uh, replicate that, but that doesn't mean it doesn't happen. Um, he saw an HNE effect uh, on some enzymes in uh, the preclinical AD,
Huh.
and but we couldn't see that. But nevertheless,
So the
this
preclinical AD that so here in Hopkins, well, at Baltimore, the NIA has its Baltimore longitudinal study of aging.
right,
So what they do is they follow people uh, beginning when whatever they're 40, 50 years old and follow them for decades and decades, and then they can go back and person X may develop Alzheimer's disease, but then they have all this data,
yeah,
they have blood samples, cerebrospinal fluid in some cases.
and
Yeah.
they can see how early it happened.
Yeah, how
Yeah,
early.
absolutely right. And lots of people have noticed that. Think in using exosomes to
Well, Dimitros Papagiannis, who was a neurologist in my lab,
yeah,
he he's he got tenure based on his work in isolating these vesicles that he thinks are coming from neurons and
right,
then showing they have uh, insulin resistance
correct.
and they have A beta and tau
Right.
pathological and
Absolutely right. So I've also been working on Down syndrome to some
uh,
extent, and you see the same process in that kind of thing. And as you know, Alzheimer, like neuropathology and
no,
dementia, occur at very early compared to an Alzheimer
yeah,
patient, typically. Um, so anyway, the issue is free radical damage to many proteins. Many of them involve transporters, including glutamate transporter that takes glutamate from the presynaptic to postsynaptic synapse. That glutamate accumulation there would normally be taken up by EAAT2,
yeah,
and sometimes it's called GLT1,
yeah,
but we're the first to show that that has HNE all over it.
yeah.
And now, hesitate to say the name of the person who said that this was inactive in uh, AD, but we think it's right in spite of his recent uh,
Huh.
demotion from NIA. And so
No, oh, yeah.
you know what I'm talking about.
Yeah, I know what you're talking about.
Yeah.
Yeah.
So
No, but we showed the same thing, right? I mean,
yeah,
and we
yeah,
showed glutamate transport's impaired in cultured
exactly.
astrocytes and
Exactly.
yeah,
So he said specifically this EAAT2 was oxidized, not oxidized, was dysfunctional. And what we say is now we can explain that
yeah,
by H&E binding to this transporter, changing its function and therefore not acting as like it should. And then you build up glutamate and you get excitotoxicity or
yeah,
something like that happening
yeah.
uh, through the glutamate
And
receptor.
that also makes sense. Alzheimer's patients have like a 30 to 50 fold increased incidence of seizures.
Yeah,
And
that's right.
recording EEG, a lot of times Alzheimer's patients, they may not have a seizure, but if you look at their EEG, they have kind of what you'd say pre-seizure hyperactivity.
I see. Yeah, very good. So anyway, what we see, what we're suggesting is there's a disruption of uh, metabolism
Yeah.
through glucose primarily, but other ways too, and a disruption of calcium. You certainly are one of the pioneers of that field, and a disruption of other signaling pathways that are normally needed for the survival of cells. And these key proteins are themselves oxidatively damaged. So oxidative damage, in our view, is fundamental to the development of MCI and AD.
Yeah.
And that is the story. And in fact, I think um, certainly your work, but ours too, has contributed to the idea of changing the paradigm of diagnosing or saying causation of these diseases of MCI and AD, that you just have to now include oxidative damage as part of the story.
Yeah.
It'd be disingenuous not to,
Yeah.
uh, because here we explain lots of ways in which neuronal death happens that emanates from oxidative damage. And if you remove certain components of A beta. And by the way, we argue that A beta is pretty essential to that uh, as a small oligomer, but not a big oligomer. Uh, Dennis Sulko showed that synapses don't work with large oligomers. I'm sorry, they are not affected by large
Yeah.
oligomers,
Yeah. Yeah.
but by small oligomers, they block the action of synapses. And that would fit exactly what we've defined. Yeah, some of the proteins we've defined have to do with neurotransmission. And
Oh, yeah. And I mean, the degeneration begins at the synapses. There's no,
you're the one that first you invented synaptosis as a
yeah.
name. And absolutely, I couldn't agree more. That's exactly right. But anyway, we have found a lot of proteins that are involved in, you know, docking a vesicle to the presynaptic membrane don't work, or recycling vesicles back to the presynaptic neuron don't work, and because they're oxidized, and it's really important process.
And I think we're both on the same wavelength as far, once someone's diagnosed with Alzheimer's disease, there's already a lot of damage in their brain.
Huge. Yeah,
And so any approach that's based on uh, ameliorating the oxidative stress or the consequences, conse I think we have to think about uh, risk reduction,
right,
prevention,
right.
rather than I'm very skeptical that any antioxidant is going to have much effect once a person's already.
Yeah, the damage is done. Yeah, that exactly. Uh, Now, I think so. We went back to the beagle dog, and
Okay.
we fed the beagle dog at age 12. Now, when I say we, I don't mean me. I mean our colleagues in California. They they fed the dogs a high antioxidant diet, and they performed or not. So regular diet, high antioxidant diet, and then a experiential uh, time learning new things.
Ah, yeah, I remember. I
12-year-old
remember those. Yeah.
and or not. And what we found with a fellow who did a PhD with me, then later, by the way, got an MD at UK and is now he was chief resident, but now he's a practicing physician at Central Baptist Hospital in Lexington.
Huh.
Um, anyway, he showed that the reason that high antioxidant diet and behavioral enrichment worked was to uh, reduce the enzymes associated with the very processes you and I have just been talking about. calcium, neurotransmission, other sorts of things to reduce the oxidation in those entities compared to the others. So I tell my students about, in my neurochemistry class that I invented at UK, I ask when I show that result, I say to them, remember, just all through your life, you need to be eating food that is healthy for you. And like broccoli, which has in it a compound that can lead to the activation of NRF2, which is uh, a transcription factor that helps the production of new antioxidant enzymes.
Yeah.
And they're quite potent. And one of them is heme oxygenase, which we mentioned before, leads to biliverdin reductase. And
There's another one that actually acts in the membrane, NQO1,
yeah, that's true.
the quinone oxidoreductase.
Yeah. And that would have direct influence on the Q cycle.
Yeah.
So that's for sure. Um, and so my recommendation are to do basically three things. One is eat reasonably. Now, and with let's see, there was a fellow who wrote a book here a while ago about, he said, uh, food, mostly plants, and not so much fat related food.
Was it Dan Buettner, the Blue Zone guy?
That doesn't sound right. But
Mm.
anyway, um, the idea,
Oh, I know who it was. Michael Pollan.
yes, Michael
Yeah.
Pollan. Yeah, he said food, mostly plants,
Yeah.
and
And
colored foods.
and uh, moderation in calorie intake,
Yeah, moderation
maybe a
of
little
calories.
bit of intermittent fasting.
Well, yeah, that part is going to be harder for most people to do, but
No, it's not. It takes several weeks or a month to adapt.
yeah.
Yeah.
Well, in any case, you've definitely shown that that's helpful to you. And I think that the let's
I
see,
don't
so,
know. In a randomized, in a randomized controlled trials that I've been involved with and many others, it's the people can stick with it if they can get through the first several weeks to a month.
okay,
And we know why. We know what's going on in the neuroendocrine systems that control hunger, appetite,
right,
and it takes that time for those to change.
adapt, yeah,
Yeah.
adapt.
Yeah.
Um, so, uh, the three things:
Yeah.
diet, related to what we've just been talking about, exercise if you can. Doesn't have to be great exercise. It can be just walking.
Yeah.
Turns out you don't have to be just beating the heck out of yourself. You can just walk.
Yeah.
The idea is blood flow.
Yeah.
Move blood along, I think. And the third thing is stimulate your cognitive senses. In other words, challenge yourself to learn something new. And what I tell people is I have a lot of collaborations of people who did their postdocs with me or PhDs with me who are now full professors at the University of Rome and Sapienza University of Rome. And
So listen to the Brain Ponderings podcast regularly,
yeah, there
and
you go.
then read up on the.
Yeah. So anyway, the idea is to uh, stimulate yourself by challenging yourself. So I say to people, well, I have learned just enough Italian to be dangerous. So I, though the verb tenses are awful, so I can't figure those out. But at least when I'm in Italy, I can figure out what train to take and where the bathrooms are and how to order food. So,
That's the same for me with Spanish.
yeah,
Yeah.
Yeah, well, I can understand that. But anyway, learning new things. Some people want to learn to play an instrument when they're older and as they're getting older. The point is if you can force yourself to make new synapses, then you are going to be in good shape, better shape. The so-called cognitive reserve, I believe, is a real concept and a real thing.
Yeah.
The more synapses you have, because everybody getting older is going to lose synapses. And I'm just months away from turning 80, and I feel like I'm 50, but except for my lousy knees.
Yes,
I,
yesterday there was a guy from the Netherlands that came with, and he and his colleagues are making a documentary, a multi-part documentary on depression. Okay, and so, I was talking to him about mechanisms and stuff, and there's been these nice studies showing that someone who has, severe clinical depression, and then they recover, the size of the hippocampus can increase when they recover from the depression.
wow,
So there's reversible shrinkage that occurs with depression. And that's very encouraging for people with depression, but it's also very encouraging for anybody.
no kidding.
Yeah.
Yeah, that's a great thing to hear. I knew about the studies that people had looked at violinists, for example,
Ah,
and they find different parts of their brain that control their finger movements
yes.
are
More.
much larger
Yeah.
than the typical person.
And the famous cab driver study in London where the
Right.
cab drivers had bigger hippocampus, which is very important for spatial navigation.
Yes, absolutely.
Yeah.
Yep.
Yeah.
I remember that.
Uh, so
So
that's
anyway,
good.
those three things are
That's a good
cool.
reason not to use GPS.
Exactly.
Don't use your GPS.
Because now you force yourself to think.
Memorize the map before you go.
There you go. So, in any case, having those three things, I think, are reasonable uh, approaches that, if they don't prevent the disease, I'm talking about Alzheimer now, at least they may slow either the onset or the progression.
Yeah.
And I think that could be safe in saying that. And the truth is, I mean, there isn't any real great treatment, even with these new drugs, because of, brain bleeds and other things that can happen. So, uh,
So the common mechanism here with physical exercise,
ab-
diet with a lot of plants, phytochemicals,
right,
and
right.
keeping your mind active, that all of those are uh, imposing, I think, a mild challenge, mild stress, and the cells are responding adaptively.
Exactly right.
Yeah.
It's exactly right. Professional athletes, a baseball player, a basketball player, they develop muscles and they develop skills of vision and how to see things that other people would have a hard time doing. But part of that is repetitive motion and
Yeah.
repetitive work, and you kind of do that. Well, the same thing is here. If we challenge ourselves, just like you said, if we challenge ourselves, our bodies will respond to help mitigate that challenge, and that means they will do protective things for us. And I can't agree with you anymore. I mean, that is absolutely what I think is the right approach. And now, if you didn't do that, obviously, nobody should be smoking anything
Right.
ever. So, that
Well, I guess if you smoke enough, you won't live long enough to get Alzheimer's.
well, probably, but that's a poor trade-off in my
Yeah.
view. So anyway, the idea is just as we've said, and I think. Uh, even though my lab is now closed and things are settled down, I've trained 70 PhDs and master students and 180 undergrads and about 30 postdocs. And so there's a new generation of Butterfield type people out there somewhere who are trying to carry on this idea, and I'm very happy about that. And we're hopeful. As I'm anyway, closing down the lab, I'm still just like you, trying to stay active intellectually and reviewing papers for people, for doing, for different journals, and as long as they're decent journals, and for keeping writing our own paper. I still have data of probably 6 or 7 more papers I've got to write pretty soon, and I haven't done that yet and staying up with the literature. So now I will go to your podcast, and that that will be good. I will keep learning things
Well,
that
you
way.
can, the thing to do is listen to them like on Spotify. I don't know if you've ever used Spotify.
No, but I can learn.
Just put on your cell phone. You can listen while you're walking on the trails in
There you go.
Maine.
Yeah, that's what we do. We do a lot of that.
Yeah.
And Marcia, my wife, she even does a lot of snowshoeing. And
Cool.
my knees are not good enough for snowshoeing, but she loves it. And our we have this 5.5 acre land, and there are lots of trees and lots of paths, and so she can snowshoe to her heart's
That's
content.
great exercise. Yeah.
Oh yeah, sure enough is. And of course, we're only by the crow flies, we're only 40 miles from the ocean. And so we can get to the coast fairly quickly and enjoy the ocean.
Yeah.
And with our camp just a mile away from here, we have a dock that goes 30 feet out into this big lake.
Oh, nice.
So it's fishing and swimming and that sort of thing that we really, really enjoy
Well, in the summer,
in the summer. Oh yeah, in the summer.
like you could do ice fishing, I guess.
People do,
Yeah.
and sometimes they get a little careless and
Ah,
go in through the ice, but uh, uh, that happens too often, actually. But that's usually associated with heavy drinking,
yeah.
not thinking very well. But in any case, the opportunities in Maine in this very large state with only a little over 1 million people in it. Now, in the summertime, that population triples
Yeah.
because this
Yeah.
is a big destination for vacations.
Yeah.
And in fact, the nickname on the license plate says Vacation Land, is that for Maine? So they're trying to draw more tourists as it is. Anyway,
Okay.
for a few months in Maine, it's tough driving because of all the cars. But outside of that, it's just a wonderful, wonderful place. And of course, as you said, I was born and reared in Maine, up in the woods,
Yeah.
and in a county bigger than the state of Connecticut, but with only 10,000 people in it. So
Well,
lots of space.
we still have a farm in Minnesota, 80
Yeah.
acres.
Oh, that's fantastic.
My daughter's, sorry, my sister's daughter and her family live there now, and then the neighbors farm the land, but so that land's still there.
That's fantastic, Mark. Yes, fantastic. I remember you racing your horses. You
Yeah.
and your dad came in
Yeah.
Lexington.
Still got, these are bits from our best horses.
Oh my gosh.
That's my grandpa up there,
Wow.
that black and white. Yeah.
So you've
And
carried that tradition
that's
on.
I don't know if you can see this. See that painting?
Yes.
That Bill Marksberry gave me that as a going away present.
Oh, I remember that.
And that's from the Red Mile. It's the
Yeah, correct. Yeah,
famous trail. We had a horse there the first few years I was in Kentucky.
yeah, yeah. I was impressed as heck that you were driving the sulky, is that what they're called? Sulky,
Yeah, yeah.
and uh, and is amazing. I thought, oh man, the guy's in the lab all the time, and he still has time for this, and he does a great job at that. Oh, that
Well, I didn't.
everybody's envious.
We paid someone to train the horse during the week, and then the weekend I went in for a few hours.
Yeah.
Yeah.
Well, wonderful job, and uh, I'm really happy for you in your retirement and what you're trying to do, and to stay active because that's what we have to do.
Yeah. Okay. Likewise, let's stay in touch.
My great pleasure to do so. And thank you, Mark, for inviting me to be part of this podcast. I've really enjoyed it.
I enjoyed it a lot too. I wish we could talk for another 3 hours.
Me too.
Yeah.
All the
Bye.
best.
Bye.
Bye.